So let's do a little overview of that link reaction. It's occurring in the inner matrix of the mitochondria, which is the very inside both layers of membranes. We're using up 2-pyruvate, which are the 2,3 carbon molecules out of a broken apart sugar. We're using 2 NAD pluses and 2 coenzyme A's. So that means we're going to breathe out 2 carbon dioxides. We're going to end up reducing those NAD pluses to 2 NADHs, and we end up with 2 acetyl coenzyme A's. So our pyruvate gets oxidized and our NAD plus gets reduced. So why is the link reaction not just lumped in with glycolysis? Why don't we just consider that as like the next steps on the way forward? Like why isn't pyruvate just an intermediary? Well, for aerobic organisms, it is basically part of glycolysis. But for anaerobic organisms, they don't move past glycolysis. That 2 ATP they get there is all they're getting. And they're going to use a process of fermentation to recycle their NAD pluses. Remember, you're going to run out of NAD plus, these electron acceptors, if you're only stopping at pyruvate. So what fermentation does is it allows cells to oxidize that NADH to get back to NAD plus so they can keep reusing, get those 2 AP in glycolysis, 2 AP, 2 AP, 2 ATP, ATP, and it'll end up that pyruvate will get reduced into something else. So here you can see those 2 pyruvate end up becoming acetaldehyde, which gets converted into ethanol. So that's like alcohol fermentation. But there's lots of different pathways. In addition to alcohol fermentation, maybe you're making lactic acid. There's hundreds of different fermentation pathways, things that you can make. And so why do we love fermentation? Why is it so important? Well, maybe you love yogurt. Maybe you love ajir bread. Maybe you're a big fan of kimchi. Maybe you love kombucha. Maybe you're a cheese fan. Whatever you love to eat, those are all products of fermentation. So since we're entering the mitochondria, it's good to know what is in a mitochondria. There's an outer membrane, which is exactly what it sounds like, an outer membrane on the outside of the mitochondria. There's an inner membrane, which is highly folded on the inside of the mitochondria. Those folds on the inner membrane are called cristae. Those are those folds or bends. And the reason that's there is to increase the surface area, maximize how much membrane there is. The matrix is the very inside space, which is where we said that link reaction takes place. And then the intermembrane space, the space in between those 2 membranes in the mitochondria. So here we can see our matrix on the inside. Those folds are the cristae, and you can see how folded it is to maximize surface area. Remember, our mitochondrion is a former bacteria that used to live by itself, so it has its own DNA, and it's going to have ribosomes and things like that. And then there's an outer membrane. So in that inner compartment, the matrix of the mitochondria is where the Krebs cycle, also known as the citric acid cycle. Krebs is named after the researcher who invented it, otherwise citric acid cycle, same thing. And for each molecule of sugar, we're going to go through this cycle twice. So we call each cycle a turn of the Krebs cycle. So we'll turn it twice for every glucose. So that acetyl coenzyme A comes in, and the coenzyme drops off that acetyl group to attach it to an oxaloacetate to become citrate. So we can see why that citric acid cycle is coming in. Then, through some enzyme reactions, we're going to reduce an NAD plus breathe out carbon dioxide. So we're breaking down citrate into alpha ketoglutarate. Then we're going to release some energy in breaking apart those bonds in alpha ketoglutarate, which is going to send an electron to NAD. It's going to give the energy necessary to make an ATP, and we're going to lose another carbon dioxide. That brings us to succinate. We're going to break apart succinate and rearrange it to get rid of some electrons. 2 will go to FAD to make FADH2. Then we'll have 2 going to NAD plus to make NADH. And then we have returned to oxaloacetate, which is now ready to accept another acetyl group from acetyl coenzyme A, and we go through the circle again. So each turn is going to produce 2 carbon dioxide. It's going to produce 1 ATP, 3 NADHs, and 1 FADH2. And this cycle is a cycle. Cycle is a cycle, and it just keeps going around. And those intermediaries just keep breaking and making, breaking and making, breaking and making. And so for each acetyl group, we get 1 turn. There's 2 acetyls per glucose, so 2 turns of our cycle. And that's going to make a lot of electron shuttles that we're going to get to use. And I've been keeping track, if you've noticed, of all the carbons throughout all of this. So we lost some carbon dioxide in the link reaction. And now we're losing more carbon dioxide here. So we lost 2 already. And now we're losing 4 more. 6 carbons is what we started with in sugar. So all of that sugar you ate is getting breathed right out. Or at least the carbon is. And some of the oxygen. So that's going to bring us now to oxidative phosphorylation, which is going to occur in the cristae of our inner membrane. And this is the final couple of steps in aerobic respiration. And what it does is it uses the energy from those electron shuttles to build a concentration gradient. And then it's going to use diffusion in that concentration gradient to power an ATP producing machine. So the first step is something called the electron transport chain. And if you don't learn it now, you'll learn it in photosynthesis because the same thing happens there too. So all those electrons that we've been storing up in all our NADs and all our FADs, we are now going to release. And these are high potential energy electrons. And they're going to flow through that inner membrane. And it's going to flow through these protein complexes. And that's going to give the energy necessary to push protons or hydrogen ions against the concentration gradient. So this is going to be active transport, where we're pushing from low to high concentration. And we're filling this inner membrane space up with hydrogen ions. At the end of these electron journeys, meets some oxygen that is going to combine with some of these hydrogen ions and form water. So there's where we get our water product. So why does this work? Well, think of a battery. Batteries generate electricity by completing a circuit where those electrons flow from the negative end to the positive end. And basically, this is what's happening in your membrane. The electron transport chain creates a flow of electrons that's releasing some of that high potential energy and turning that into kinetic energy, where we're pushing hydrogens against the concentration gradient and filling that inner membrane with hydrogen ions. So we have all these protein complexes. And they're just numbered 1, 2, 3, and 4. And they work with these proteins called cytochromes that are funneling these electrons and moving hydrogen ions in. This is our electron transport chain. And basically, it's just flowing through the proteins and increasing that electronegativity. And as the electrons move, it's going to give that energy. you're not going to need to memorize cytochrome C or the 4 protein complexes, but you should have a general idea that that flow of electrons from our NAD and FAD carriers is powering the movement of hydrogen, and then it binds to hydrogen and oxygen to form water at the end. And so you can see there's a whole number of different proteins involved, and those electrons start really high energy, and they slowly release their energy to power the movement, losing a lot of that free energy. So by the time they bind to form water, they're much lower energy electrons. So why are we doing this? Why are we jam-packing all these hydrogen ions in there? Well, the answer is something called chemiosmosis. And this is where we use a protein called ATP synthase, and all those hydrogen ions are going to flow through ATP synthase. And they're going to flow through it because of diffusion. So no energy is needed here. Hydrogen ions are looking to go from that high concentration intermembrane space to the low concentration matrix. So they're looking to push through. So they're just going to flow due to diffusion. And you can think of ATP synthase kind of like a rotor or a turbine. And it actually spins. It's actually moving inside your cells. And what it's doing is as those hydrogen ions flow through, it's creating that movement, which is going to create energy. And that energy that's being released is going to catalyze that formation of ATP from ADP and a free phosphate. So that ATP and phosphate, it's going to use that energy of the spinning rotor to make ATP. And really, genuinely, think of it like a hydroelectric dam. Think of it like a powerful wind turbine. And those hydrogen ions are the wind moving that turbine, or the water flowing through the dam. And that's powering the rotors to generate electricity. So as you can see in this diagram here, it's as it moves through, that's actually spinning. And that's generating that energy to power that ADP and the phosphate to form ATP. So in review, this is at our inner membrane. It's using oxygen, NAD, the 10 NADHs we've picked up, 2 from glycolysis, 2 from the links, 6 from Krebs, 2 FADH2s that we got in the Krebs cycle, and we're going to make water. We're going to get a bunch of NAD and FAD that have been oxidized to go accept electrons again, and somewhere over 30 ATP. So why is it not exact? Well, we can't give an exact number because ATP synthase and oxidation is not like directly coupled. They work together, but it's not a complete science. And so on average, from the energy we get from an NADH, from pushing those hydrogens across, we get about 3 ATP per NADH and about 2 per FADH. Now, this is definitely way more than we're going to get in anaerobic fermentation, but we don't know the exact number. In perfect, ideal, everything works perfectly, no energy lost situations, because we have 10 NADH and 2 FADH, 3 per, 2 per, if we multiply that out, you would get 10 times 3, 2 times 2, 34 ATP, plus the 2 we got in the Krebs, the 2 we got in glycolysis, and so we'll max out around 38, so just shy of 40 ATP per sugar. Now, obviously, sometimes you'll get a little less per sugar, maybe you get 32, 34, 36, and you'll see different textbooks give different numbers, but the maximum ideal, everything's going perfect conditions, will get us 38 ATP. Now, you may think, how efficient is that? Well, if we consider that 38 ATP is 19 times more efficient than anaerobic respiration, we're doing so much better than bacteria. And so your body is about 40% efficient. If you take all the energy that should be in a glucose molecule and convert that into ATP, you have about 40% efficiency, which you may think like 40%, that's a failing grade. Well, a car, if you took all the energy that's possible in gasoline and converted into what a car can convert, it's about 15% efficiency. So you are actually way more efficient than a car. Pretty cool. So in summary, cellular respiration, while it breaks down into this nice simple equation, is a little more complex. We have glycolysis, where glucose is used and some ATP is produced. We have our link reaction, if we're going to the Krebs cycle, which carbon dioxide will be produced and a little bit of ATP is produced. And then finally, oxidative phosphorylation, where the electron transport chain, we use up our oxygen, we produce water, and in chemiosmosis, we make a ton of ATP. If we're not using oxygen right after glycolysis, we're just going to recycle those NADs and FADs and keep doing glycolysis. So sugar isn't the only thing that can power this pathway. Lipids, as in fats, can be broken down and they'll enter after glycolysis. So like the glycerols will enter in for that glyceraldehyde phase that becomes pyruvate. The fatty acid chains can be involved in making acetyl coenzyme A. Proteins can get broken down to make pyruvate, acetyl coenzyme A, and join the citric acid cycle. But they're less efficient than carbohydrates, which is why we don't use them. Fats, if you burn off fat, it builds up ketones in your blood, which can turn your blood acidic, which while that sounds wicked cool, like a heavy metal song, acid for blood is actually super bad for your brain and can cause you to slip into a coma and die. So I wouldn't recommend it. And same thing with proteins. You're just not going to have the efficiency that you have with sugars. But they do work. So that is the end of my talk on cellular respiration. Hopefully it was useful, and we'll see you next time for photosynthesis.